Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
Observations and commentary on the JCO article, Adjuvant Ganglioside GM2 KLH QS21 vaccination versus observation after resection of melanoma in stage 2 patients. Results of the EORTC 18961 randomized phase 3 trial by Egermont et al. My name is John M. Kirkwood, and I am Professor of Medicine, Dermatology, and Translational Science at the University of Pittsburgh Cancer Institute in Pittsburgh, Pennsylvania. My oncologic specialty is melanoma, and I present this podcast on behalf of myself and my colleague, Lisa H. Butterfield, who is Associate Professor of Medicine, Surgery, and Immunology, and the Director of the UPCI Immunologic Monitoring and Cellular Products Laboratory. Vaccines have been a major focus of interest for melanoma investigations over the past century, and the sophistication of vaccine technology and the tools for assessment of relevant immunological endpoints have advanced enormously. In the words of the late icon of cancer immunology, Lloyd Old, to vaccinate, one must be able to effectively immunize. And in this regard, it is critical that we distinguish among a range of peptide and protein, as well as dendritic cell-based vaccines that incorporate peptides, proteins, and genetic constructs. It is also important to distinguish between fundamentally different immunological mechanisms, as well as intermediate endpoints relating to the antigen-specific CD8 killer T-cell and CD4 helper T-cell responses as compared with ganglioside and carbohydrate vaccines that elicit humoral responses that can be readily assessed using serological tests for antibody. Added to the complexity of the widening variety of vaccines that are in current clinical trials for human cancers, and the increasing sophistication of our tools for the assessment of the immune response underlying these vaccine interventions, the elements of the trial design and reporting are worth discussing in relation to EORTC 18961 as reported in the recent issue of JCO. The vaccine GMK is comprised of a ganglioside GM2 coupled with the carrier protein KLH and a strong saponin immunological adjuvant QS21. This is a commercial evolution of the ganglioside vaccine first reported by Livingston et al. In an early single-institution randomized study of a formulation with extracted GM2 to induce IgM and IgG immune responses indicating immunoglobulin class switching and associated with improved relapse-free survival, although the small size of this initial trial was only hypothesis generating. This spurred the development of the U.S. intergroup trial E1694, in which the GMK vaccine was compared against the FDA-approved reference standard of high-dose interferon alpha-2b in a trial that focused upon high-risk operable AJCC stage 2b, 3a, and 3b disease groups. That study was closed for early evidence of GMK inferiority to interferon, with relapses and deaths that were higher on the GMK arm, with a hazard ratio of 1.5 for the GMK versus interferon. Corollary immunological assessments of the E1694 trial demonstrated that the leporeldian extracted GM2 tested in this formulation of GMK induced effective immunization with IgM responses among 90% of patients by day 29 and IgG responses in half of patients at the end of 1 year. Against this background, the current and largest study of GMK is notable since it focused upon stage 2 patients of lower recurrence risk and compared a different GN2-containing vaccine against observation, with results that have been presented twice previously to ASCO. The study was presented first at closure for futility when 91 deaths among 1,314 patients, that is 6.9%, showed a 3% differential, that is to say 21, favoring observation. This difference was likely due to small numbers, although the concern that the vaccine might have enhanced relapse or mortality rates was not borne out at the final analysis presented to ASCO and detailed in this publication. This study shows no differences in relapse frequency or mortality with the GMK vaccine, and the only difference noted, which was not based upon planned analysis, is the transient and marginally significant diminished survival from first relapse among GMK recipients. When trials are negative for planned outcomes of relapse-free and overall survival, the dredging for differences in unplanned secondary analysis is always dangerous. And for this trial, in which a large number of subjects were enrolled, the unplanned analysis is further complicated by the lack of any immunological corollary endpoints as studied to advantage earlier by Livingston in the Phase 2 trial that initially prompted the larger study of ECOG and the U.S. intergroup as well as the present study. Did this study of a more recent formulation of GM2 show no benefit because it did not immunize, or because immunization against GM2 is ineffective in stage 2 melanoma patients? Unfortunately, the lack of any immunological corollary studies in this large study leaves us without any information in regard to the immunological response of stage 2 patients to the vaccine, which if present would refute the study hypothesis, or alternatively, if absent, would signify the failure of the intervention to effectively immunize the stage 2 patients. The post-relapse survival disadvantage noted by the investigators of this EORTC trial are more problematic since relapse detection for the early disease population receiving GMK vaccination here may have been detected earlier. That is a lead time bias, making the apparent post-relapse disadvantage of the GMK group artifactual, since this trial did not specify rigorous relapse surveillance as would have been required for relapse detection if this were a primary regulatory endpoint. What hypothesis might have been advanced to explain this post-relapse disadvantage if it were real? Why would the difference be transient in years 1 to 4 and then dissipate? Again, the lack of any immunological corollary data for this intervention makes it difficult to come to any conclusions. The more important issues surround the place of stage 2 melanoma patients in the investigation of immunotherapy, where the hypothesis has been that earlier disease may be more susceptible to immunotherapy interventions, and this would be due to potentially greater immunologic responsiveness or not yet fully engaged tumor microenvironment immunosuppression. And time for full evolution of broad and robust anti-tumor immunity may be greater. On the other hand, there is concern that the lesser tumor burden of stage 2 patients may provide a lesser antigenic exposure and therefore a lesser likelihood of benefit from cytokines, interferons, and checkpoint blockade interventions. Indeed, it is now a time when trials of anti-PD-1 and PD-L1 aimed at higher-risk groups, stage 3B and stage 4, are being developed. Given the toxicity of first-generation CTLA-4 blocking antibodies such as ipilimumab and tremelimumab, it is not surprising that these trials have skirted the lower-risk stage 2 group, and at this point, anti-PD-1 has greater efficacy and lesser toxicity, prompting consideration of the stage 2A-B groups as reasonable forums for investigation of these modalities. The hypothesis here would be that the differential effect may be even greater in this population than in the populations of higher disease burden. For vaccines in particular, this large subpopulation of patients is reasonable to continue to explore with the hypothesis that it may be more likely to benefit from specific vaccine interventions, generating tumor antigen-specific memory responses. The stage 2 forum for melanoma is a large one, and due to this numeric peremptory, it has been the focus of recent ECOG-led intergroup trials, such as E1697, which were presented ASCO several years ago. That study showed only transient early suggestions of benefit from 1 month of interferon therapy compared against observation and was ultimately closed for futility as well. In each of our current trials for melanoma, and in particular for adjuvant therapies where the clinical impact upon disease relapse, mortality, and post-relapse effects are to be considered, it is mandatory that corollary mechanistic assessment be included to better interpret the results. The corollary studies we have performed in the context of the neoadjuvant interferon and ipilimumab agents will give way to studies of even more active immunomodulators and combination of these agents where the ultimate goal will be to enhance vaccine immune responses. The assessment of specific immune response, such as antibody, T-cell immune effectors, and other parameters, will need to be assessed in order to make sense of these outcomes. We look forward to the more rigorous studies of clinical endpoints such as relapse and mortality and more informative correlates, both immunological and molecular, to accelerate progress in this field. This concludes this JCO podcast. I thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.